So, dear participants, now this is the time for the second talk. And since it's a virtual program, so you can, while listening also, you can take a break. You can have coffee, tea, something in your home itself. Because I know this is a very difficult, tedious time. Because the transition time for many, it is also a kind of in home also, there is lots of engagement. But still, we have 100 participants. So, I welcome now Dr. Amol Solanke, a very well-known scientist in the area of plant biotechnology. Dr. Amol Solanke is a senior scientist in ICR, National Institute of Plant Biotechnology, New Delhi. And in fact, I must say that we work together for a national program in Delhi that covers the entire country, certification program. And at that time also, I got a excellent cooperation from him for managing that important activity. I think for 15 years, 2006 to 2021, I did that program, and Dr. Solanke was very important part from the referral center for managing the referral samples, developing protocol, validation, new kit, new protocol, and providing training. Dr. Solanke's PhD from Delhi University on plant molecular biology, and his MSc and BSc from Maharashtra, MPKV Rahuri, and PDKV Akola, respectively. He has received many awards. List will go very long. He is distinct scientist to Purdue University, USA, Jawaharlal Nehru Awards, NASA Fellow, National Environmental Science Academy, CV Jacob Awards, CSR, JRF, many. I'm not reading out those things. List will go very long. That proves that how much he has contributed for science in a society. He has filed 2 patents, 100 plus publications, many book chapters, 15 plus, and many publication in H Index and I10. Many books. He was also involved in HRD training program, and 50 plus invited talk he has delivered in global and a national platform. So with this strong background, he is here to talk about plant molecular biology for sustainable agriculture, sustainable development. A very important talk for the biotechnologist, plant biotechnologist, agriculture scientist. So I welcome Dr. Solanke once again. Over to you.
Thank you very much, sir. Am I audible?
Yes, we can hear you.
Okay, thank you for such a nice introduction, sir. Here actually, just a minute, yeah. Okay, I hope my slides are visible.
Visible.
Okay, so thank you very much, sir. It was very nice to work with you. Actually, you were mentor in that program, and you take us with you, and really that particular program help a lot for India. And I have few slides about that program also in my this molecular approaches.
Oh, nice.
Okay. So thank you very much for giving me this opportunity and giving me this particular topic, because I feel this is very relevant and very close to my heart also, and my everyday working what I am doing. So this is molecular approaches for sustainable agriculture. So in this case, I will mostly talk about molecular approaches which we are using for sustainable agriculture. And I know since this is 4th day of this particular program, lot has been talk about this sustainable agriculture, but I want to discuss here what are the factors which affect this sustainability of the agriculture. So if we talk about history of agriculture, so it was with trial and error, almost I will say 9,900 years back. After that, we have used these scientific principles of breeding from last 120 years after Mendel. And then we got this chemical induced mutation, and we thought that we will change agriculture with anything, but it does not happen. And then 30 years back, we witnessed this recombination DNA technology, and we got many results from that, like we are enjoying BT cotton in India. And then the era of genomics and bioinformatics appeared 20 years back. And recently, just 10 years back, we have seen this genome editing. So this is a small, we will say, life timeline for this one, from domestication or Mendel or mutation or marker state selection. Now we have reached to genome editing. But if we see our crops, so we have seen that whatever crops we are using, those were not actually crops in the beginning. So teosinte, if we see, we cannot eat this, but we have changed this by domestication and selection. Or this small tomato, this is 1 centimeter tomato, and now we can see the larger tomato. Again, this is because of, we will say, selection and domestication that we have seen. And if we go just recent back, our agriculture was so proliferating across the world. So we were in the international trade with our cotton, indigo, opium, and rice. And we have irrigation system that was before, we will say, acquiring of this British to India. And then we have seen that after acquiring this British, they have established Department of Revenue and Agriculture, and we have started systematic agriculture there. And after that, in 1903, this Imperial Agricultural Research Institute was established in Pusa, Bihar. But unfortunately, in 1936, there was a huge earthquake, and after that, it was shifted to Delhi. And right now, where I am sitting, that is Indian Agricultural Research Institute. It is actually coming from Bihar. That place was called Pusa, and that's why we called it as a Pusa Institute right now. So after that, in 1926, Royal Commission of Agriculture was established, and from 1891 to 1946, agriculture was deteriorating in India, and because of which the situation was very pathetic because of this Bengal famine. Okay, I know for many people these pictures are disturbing. Okay, like if we see this particular picture, the person was sleeping somewhere in the field, and there itself it died and no one was there. So this was most devastating era, 1943, which is Bengal famine, and where agriculture production was declined to 0.7%, and almost 2 million, just imagine at that time, there must be, I will say, 20 or 30 crore people, and out of that, 2 million people died because of food scarcity, 1 million cattle died because of this one, and almost 4 million people's life get deteriorated. And from this onwards, we got independent in 1947, and after that, the condition was not so good. But with the help of Norman Borlaug and MS Swaminathan, we have witnessed Green Revolution. And again, from this point onwards, these molecular approaches entered in the agriculture. So there were dwarfing genes. So along with dwarfing genes, these two gems with irrigation facility, improved and hybrid seed, chemical fertilizer, pest management, farm credit, and political will. We should not forget this political will of that time. we have witnessed green revolution, and it was not only in India, it was across nation. Many nations benefited by green revolution in wheat and rice. And after that, we have seen as India here rainbow evolution, where food grain production increased fivefold, milk production increased eightfold, fish production increased 11-fold, horticulture production altogether increased sixfold, meat production eightfold, egg production 27-fold, and poverty and hunger percentage was half as compared to earlier one. And again, this happened when we have just 2% of world's land, 4% of fresh water, and with 16% of world's population. But is this enough for India, country like India? No. Because now, this year onwards, we are top in the population. So our population is increasing, and it is expected to be 1,620 million in 2051. And again, most important is it is shifting from rural area to urban area. So it means people are moving from agriculture. Again, if we see this particular, we are not able to meet with our increased food production with this increased population. And if I talk about global hunger index, this is a really pathetic condition. Okay, out of 127 countries, our number is 111. Even our neighbors like Pakistan, Bangladesh, Nepal, and Sri Lanka have better global hunger index. We have witnessed green revolution, but because of this, the pattern of crops get changed. And mostly if we see in Asia, we are mostly depend on these three crops, rice, wheat, and maize. Whereas if we see in Africa, almost all crops have their presence in their food. Again, we are tilting towards very few crops like soybean, palm oil, sunflower, wheat, rapeseed, mustard, rice, and there is decline of these crops like sorghum, cassava, millets, coconut. So it is reducing. So slowly it is narrowing down towards very few crops. And this is not good. If we see this particular, we will say, number of crops from which we are meeting our, we will say, daily needs like calories, fats, protein, or weight. So if we see in 1961, there were larger number of food material available with us, or I will say diversity in food. Then in 85, it reduced down. And in 2009, if we see, there are very few. So what you eat in the morning, either wheat or rice or sometimes maize and few vegetables. So the diversity in our food supply get reduced. And it is not only for this one, it is for everything, even calories from limited crops, fat from limited crops, protein from limited crops. Then another challenge is climate change. So if we see here, temperature is going to increase, and because of which there is alarming condition in country like India. Again, availability of water is going to reduce by 40%, and because of which crop production is going to reduce, and that we have already witnessing in Rajasthan or even in Punjab. Okay, even if we see fertilizer use efficiency, during 1980, from 1 kg of nitrogen, we were getting up to 60 kg of grain. But now it has been reduced to just 15 kg. Again, if we see from 1950, in our soil, there is nutrition deficiency of many we will say micronutrients and macronutrients. And again, there is decline of crop area because of, we will say, this concrete jungle, or which can or other things for which we are utilizing the agriculture land. Again, there is problem with biotic and abiotic stresses, which are as compared to its actual yield, average yield, we will say these biotic and abiotic stresses are taking toll of many of these crops. So altogether, I will say these are the challenges in front of our agriculture. That is population, which is going to increase by 1.6 billion by 2050. The shrinking area of cultivated land, diminishing water resources, malnutrition and undernutrition, deterioration of soil, and then climate change. Okay, and if we see here, the role of molecular approaches is going to play important role by raising the yield ceiling, which can provide sustainable production. So thus, resource-based, science-based and information-based can lead to next generation sustainable agriculture. Okay, so if we talk about definition of sustainable agriculture, okay, this is the long definition given by FAO in 1988, but further International Union of Conservation of Nature, they have simplified it as a transform our complex food production or consumption system so that they do not degrade the biodiversity and ecosystem services on which they are depending. Okay, so these are five principles on which sustainable agriculture is there. That is improving efficiency in the use of resources, conserving, protecting and enhancing natural ecosystem, protecting and improving rural livelihood and social well-being, enhancing the resilience of people, communities and ecosystem, promoting good governance of both natural and human system. Okay, and if we talk about genetic improvement, there are few approaches like selection, domestication, base broadening, and new breeding techniques and gene modification through biotechnology. So in this case, already these three we are using from many years. By selection, we are developing, we are selecting better crops. We are domesticating new crops. We are by crossing, we are broadening the base of whatever genetic material is with us. But I will mostly talk about this last one, that is new breeding techniques and genetic modification through biotechnology. Sir?
Yeah.
Yeah, do we need to take some break for questions in between? Or at the end there will be?
We can take at the end. It is up to you.
So if someone have query, I think in between, they can ask, we can take small breaks in between so that there will be proper continuity and your involvement.
Perfect.
So if no question, okay, so when we are talking about molecular approaches, which molecules we are talking about? So in this case, we are going to talk about genes, we are talking about transcriptome or transcript, we are talking about proteins, or we are talking about metabolites, because these are the things which are actually giving phenotype. And what is that phenotype? This particular phenotype may be your higher yield, maybe you are resistant to some biotic stresses or tolerance to abiotic stresses, or maybe some new phenotypes like golden rice, I will discuss, because golden rice is not available in the nature. But with these molecular approaches, we have developed this kind of things which can benefit in sustainable manner for humankind. Okay, so mostly I am going to discuss about these molecular approaches like tissue culture industry, genomics, molecular breeding, genetic engineering, genome editing, and crops with novel traits. Okay, so there is industry called biotech industry, and which is, I will say, proliferating in India. And here, right now, India is at the 12th position across the world and third in India Pacific region. And here, if we see, this is 2% global market share of biotech industry is there. And right now there are 800 plus companies in India. And by 2025, we are expecting that this biotech industry will be of $100 billion. And this is slowly increasing. And these are the components of this particular biopharma, bioservices, bioagriculture, bioindustrial, then bioinformatics. So slowly this particular, and obviously biopharma is more proliferating as compared to other, but slowly these other industries are also taking speed. Okay, so what is this? Out of this, I am talking about this tissue culture industry. So what is tissue culture? This is the first product, biotech product, that is Pusa Jai Kisan, which is high yielding mustard variety. And this is Somaconol variant of Varuna variety. This was released in 1994 and named as a Pusa Jai Kisan. And till date, this has third position across all farmers which are growing this brassica. Okay, then there is tissue culture industry, which sir was actually heading when he was in BCIL, and this was in this particular industry. If we talk about tissue culture, it is asexual mode of propagation, which is used for multiplication of commercial tissue culture under tissue culture facilities. This can be used as a micropropagation for banana, papaya, sugarcane, cashew, or coconut, or as a clonal propagation for crops like eucalyptus or teak, or meristem culture like potato or citrus, or if we want to generate double haploid or shortening the breeding cycle. Here I want to give one example that if we grow bamboo and if it that particular flower after either 40 years or some flower we will say species after 80 years. But under tissue culture, this bamboo can be flower just after 6 months. So that is possible in this and many varieties of this bamboo are in market right now. Okay, and this is the, we will say, one particular program of DBT, National Certification System for Tissue Culture Raised Plants. And when Shukla sir was in BCIL, he was taking care of this, he was managing this particular program, and this particular program really supported tissue culture industry in India, and they have shaped this particular industry. And right now, there are 200 commercial, we will say, tissue culture companies in India, and where 500 million plantlets per annum are provided to farmers. I think Shukla sir can correct if number has been increased.
It is quite stagnant for a long time. What I could see from the website, currently, I think 93 companies are recognized.
Yes,
So,
sir.
but just to add something, so that we can take 2-3 minute break also, because
Exactly.
speaking continuously also is a very difficult task. this time. 100 companies we achieved in 2015. From 2015 to 2021, it was plus minus 5 all the time. Sometimes graph was up, sometimes less. In Project Monitoring Committee, PMEC, we reported this time. So in 2021, we submitted proposal to Department of Biotechnology that time. Then there was a committee called Apex Committee. So we had given plan for 5 years, saying that the small companies which are in southern region, Hosur region, basically Maharashtra, Tamil Nadu and Kerala. So that region covers it across 30 to 40 small companies. Even Maharashtra, we have very, very small companies. In northeastern region, companies are growing orchids. So their capacity has to be rationalized based on the crop. Because orchid is very slow growing. Banana grows very fast. So by doing this, we thought that we'll add more number of companies. So that was the plan. So I think it must be in pipeline. I don't know because I left the program in 2021. So that was the plan. So we can have 200 plus companies. Capacity can grow up to 700 million. Right now it is 500 million. The turnover, it should be at least 1,000 crores. But right now it is mainly depending on the banana. And
Exactly.
mainly it is covering only one crop, which is Grannen. So I must say that companies should be promoted to use very, very different type of genotype, local varieties. In South India, we have many varieties like Nandran, Cheni Champa in Northeast. So that kind of varieties should be promoted to avoid monoculture. So though banana industry is growing and we have reported in the Pari report, what we have written, banana tissue culture, a success story. So still there is great potential. That's why we are talking in this FDP. That new variety, new crop. Yesterday, Dr. Susmita Sukla she also talked about medicinal plant, tree. So that kind of new variety has to be included, and companies should use at least 5 to 6 crop in if they want to succeed in commercial venture. Because I have seen companies failing in certain aspect because they are depending on only one crop all the time. And banana sometimes it will grow, sometimes it is down. So that will kind of create uncertainty. So this is scenario. So I would say it is stagnant for some time. So that's why we need some kind of technical support. We need to extend support. Also, subsidy is there, but alone subsidy will not help. Proper conducive environment has to be continued. And if it is continued, then definitely number will grow up to 200. We have great potential considering diversified condition, diverse geographic condition, agroclimatic condition, new variety can be grown like date palm. Date palm, it has not picked the way it was expected. So we have large belt of desert and pomegranate. Still there is great potential. Uh, Potato mini tuber, only the company which are processing, they are growing it. So still it can be increased. Oil palm, tissue culture, government is trying to promote, but still the imported varieties are coming. So there are many, many plant species that can be commercialized. There is great scope. And because of this technical manpower, we hope that slowly it will get materialized. So that was from my end. This is just an addition what you are presenting. Thank you so much. Please continue.
Yeah. Right now, along with this G9, they are also growing Ilaki, Puvan in some parts of Kerala, and especially there are diversity in bamboo. It is almost 10 to 20 different, not only varieties, but species they are growing. And it is specifically for paper. They have other varieties for some utensil preparation. They have another varieties or other things like for construction. They have another different varieties. So in some crops, at least they are increasing. And if we talk about pomegranate, its area is increasing, especially in UP, Jharkhand, even in Rajasthan. So now this Bhagwa variety of pomegranate, it is picking, especially in this Rajasthan area. So we are still there as a referral center for genetic fidelity, and our job is to develop protocol for this identification of this genetic material and identify its fidelity with its mother plant, so that farmer can get benefited, that they should get pure material which they are expecting and for which they are paying money. Then second thing which I am talking here is related to whole genome sequencing and genomics. So we know that first genome has been sequenced of human genome in 2000, and after that first crop genome was or we will say plant genome was sequenced was Arabidopsis again in 2000, but real crop genome was started sequencing in 2005, which was rice. And we were part of this particular genome sequencing program where we have sequenced large arm of chromosome 11 of this Nippon bear variety. And then we were part of this another consortium called Tomato Genome Sequencing Consortium. And I was part of this particular consortium where we have sequenced this hens variety and chromosome 5 from India. After that, when we got expertise in sequencing, we independently started sequencing crop of our own important or indigenous crop, we can say, like pigeon pea, which is mostly grown and consumed in India. So this was, if we see here, all these particular institutes and universities are from India, who were part of this particular sequencing of pigeon pea, where Asha variety has been sequenced. Then another important crop of India was mango. So where Amrapali variety, we have sequenced. This is highly, we will say, heterologous crop, where we will say, and because of which even if we see, it was very difficult to assemble this particular crop. After that, we also sequenced jute, and then finally in 2018 we sequenced wheat genome. This is because if we see the genome size of all other crops, it was in few GBs or less than even 1 GB. But here in wheat, it is 16 GB, having 3 set of genome A, B, and D, total 42 genomes. So we have sequenced this, and again in this consortium, India was part of to sequence this wheat. So right now, if we talk about any crop, it has been sequenced, and even sequencing cost is very less nowadays. This is a brief history of sequencing. I will not talk about this, but now we are not talking about single genome sequencing. We are talking about complete Earth genome. Okay, I will talk in the last slide about what is Earth genome. So whatever information we are getting from this particular genome sequencing, that we are using for molecular breeding. So like here we are linking some markers. Here I will say DNA markers or fragment of DNA with some trait. Like for example, here in this case, this is related to bacterial blight resistance. So markers are linked with these genes that is called XA13 and XA21, and these genes has been incorporated by foreground and background selection and transfer it into Pusa Basmati, and we have developed improved Pusa Basmati. So this NIPB along with genetics division of IRI, this particular improved Pusa Basmati has been developed. And if we see here, the yield increase was almost 12% as compared to Pusa Basmati and almost 33% as compared to Tarori Basmati. So in which there were two genes were incorporated by marker state selection. With same method, another mega variety was used, that was Samba Masuri. And in this mega variety, three genes were pyramided for again bacterial blight resistance, that was XA5, XA13 and XA21, and it was released in 2008. Then there is one gene called SUB1. So these are single molecules, or we will say because of which huge evolution can occur in this plant system. For example, if we see, if this particular gene is present, plant can sustain or present there in, we will say, field up to 18 days with complete flooding. Completely plants are underwater for 18 days and still it will not die. But if it is not there, this particular gene will be not there, it completely dies. Okay. So if we shift or transfer this particular, we will say QTL into any variety, high yielding variety, we can make it submergence tolerance. So this particular has been transferred. And if we see again, when we are talking about molecular approach, so this is single gene that is called ethylene responsive factor. This is transcription factor. So because of this single transcription factor, we are getting this submergence tolerant lines. This is very beautiful picture where you can see if this gene is, this is normal Samba and this is Samba SUB1, incorporation of single gene. So under submergence it is surviving, but this Samba Masuri or Samba, very few plants are surviving. So this particular is again Saltol locus, which is related to salt tolerance. So if this particular locus is present, it can sustain with high salt concentration in the soil, but otherwise normal varieties which are sensitive to salt, it will die. Then there is another QTL that is called PUP1. Okay, or which are, this is for phosphorus starvation. So under very less phosphorus also, these plants can survive and plant can give produce. So this particular has been identified from again Indian variety Kasalat. So in which this particular 90 kb indel is present only in this Kasalat, which was not present in other variety, and because of which even under very less phosphorus it can be grown. And we have seen there is very large area where there is poor, very poor or poor phosphorus is there. So it can be used in this condition. Then whatever information we are getting, those two molecules, or we will say markers, it can be put into the chip. So we have developed, as NIPB, we have developed these chips for, we will say rice, mango, pigeon pea, and using these chips, we are developing new varieties, marker state varieties. So how we are using this 50K chip? So whenever we are crossing and we are developing progeny with foreground selection, so for background selection, we can identify that there should be very less number of markers from, we will say, other donor of this particular genes which we are taking into our recipient varieties. Okay, we are also using this wild rice for pre-breeding so that these genes can be genes for different biotic and abiotic stresses can be from wild progenitor can be used by domestication into domesticate, and very few genes can be selected in these lines. So this is the list of all marker state breeding varieties in the rice. So here I have given list of only rice varieties. So there are other crops also And from last year and this year, our honorable PM has released more than 100 varieties using marker estate breeding. So this is related to marker estate breeding for quality protein maize. Okay, and now I am going to next approach, that is genetically modified and genome edited crops. Any query up to this? Okay, if no,
2-3 questions they placed.
yeah, we can discuss it in between.
Why BT brinjal is not commercialized in India?
Yeah, I am coming to this. I think this is just first slide of that particular part of my
Okay.
talk.
Golden rice available in Indian market? No.
No, sir, I am going to answer all these questions.
Okay.
So I want questions for this previous part of my talk, if it is there.
They are
Both questions are related to my next part.
precursor question they are sending.
Yes, sir.
Questions are becoming precursor for your presentation.
That is nice. Okay, so next part is genetically modified and genome edited crops. So if we see this, we call it as a biotech crop. We don't differentiate it as a genome edited crops or we will say genetically modified crops. So it is called biotech crops, and almost now 192 million hectare is under biotech crop, and there are 26 countries which are planting, and total 17 million farmers are growing these crops. And if we see out of, we will say, around 120 or 130 countries, 70 countries are adopted these biotech crops. But I am going to tell you the story of cotton. This is, I will say, this is success story which is present in India. This is the only crop which is grown in India as a, we will say, genetically modified crop. But if we see its adaptation, it is almost 95 to 100%. So whatever cotton we are growing in India, complete cotton is BT cotton. Varieties may be different, but if we see in 2001-2, the yield of cotton was just 308 kg, and now it has been increased to 550. It is not because the gene which has been incorporated, these were related to increase in yield, but it were protecting plant from this Helicoverpa arbigera, this insect which was devastating this particular crop. And the production in terms of million bales has increased from 13.6 to 37. Obviously, because of that, area get also increased. And if we see import and export, we were earlier importing these cotton bales, that was 4.5 million metric ton, and now we are exporting it as a 1.25. And if we talk about pesticide spray on cotton, it is almost 26-fold less as compared to 2001. Okay, forget about this. What is its use for sustainability of agriculture? So if we see here, 37% reduction in altogether chemical pesticide use. Then there is increase of yield by 22%. And most important thing is that there was increase in profit, cotton growing farmers. It was 68%. So this is really we will say, success story which we need to show, and we need to use other crops also in India. Okay, there is another crop which is also, we will say, in the pipeline of coming to farmers' field. That was, we will say, hybrid mustard, and where uh, they have used Barnes Basta system so that your final product will be without transgenic. So it has been, we will say, with some conditions approved by uh, government, but it is yet to see, we will say, on farmers' field. Okay, then there is BT brinjal. So here Cryogen has been incorporated, and this was under public-private partnership with MAICO, and with this particular it has been increased, but this could not release in India because of a policy matter. But same material has been released in Bangladesh, and from Bangladesh, now we are able to see that this particular BT brinjal is better. So it is better for, we will say, consumption. There is no use, but because of policy matters, we cannot use this particular thing. Okay, then there is another GM technology which saves Hawaiian, we will say, papaya industry. So in this case, there are two varieties called Rainbow and Sun Up, which were developed by overexpressing this coat protein of papaya, and because of which this is resistant. So you can see this middle patch, which is transgenic papaya, and this is non-transgenic papaya, and you can easily see the variation present here. Then there is herbicide tolerant lines. Still this particular trait is not present in India, but if we see, this is conservation tillage and it gives better weed control. It facilitates adoption of reduced tillage system, sustainable agriculture system by maintaining natural resources, enhancing nutritional availability since we are not every time plowing the soil, and less damage of crop, and because of which there is improved yield. Then through transgenics, by simply using one bacterial protein CSPB, this Monsanto company developed drought tolerant maize. Then this is another example called freeze tolerant eucalyptus. So it is not only in crop plants, it is even in, we will say, forest plants also. So if we see here, because of some genes which has been incorporated, this particular eucalyptus plant can sustain under -10 to -12 degrees Celsius, where this control plant is dying. Okay, and this is, I will say, final technique, which is actually, we will say, new plant breeding innovation, that is genome editing. This is the technique in which DNA is inserted, replaced, or removed from the genome, and it can be rapidly and economically, which is sequence-specific modification, and it is very precise in manner. And because nowadays there are many products which has been developed to this particular genome edited technique. And recently, just 4 years back, or now I will say 5 years back, these two ladies, Carpentier and Doudna, they got a Nobel Prize in chemistry for this CRISPR genome editing technology. So what is this technology? In this case, this RNA-directed DNA mutation is possible in the presence of single protein called Cas9. So like this guide RNA sits there where there is homology, and after that, by using this palm sequence, it exactly finds this position. And after that, there is double stranded break, and further naturally this particular double stranded break get replaced, or we will say repaired, and on the basis of how it get repaired, we get variations. Okay, so there are three type of variations we got, because when after this cutting by this CRISPR-Cas9 guide RNA, double-stranded DNA cuts, and when it get repaired by non-homologous end joining method, so some nucleotides may get added or few nucleotides may get deleted randomly. Okay, that is called SDN1, site-directed nuclease 1. When there is homology-directed recombination, when we add something which have homology at these ends, okay, so we replace it with two types in which there is repair, where we called SDN2. So few, we will say, variations or mutations can be there, and other where new gene can be incorporated like that of our transgenic. So in India, right now, this SGN1 and SDN2 has been approved as a without it is not considered as transgenics, and it can be there are some with some guidelines it can be released as variety. There is still there is problem, and what is there? It is single or few base edits, and it is either knockdown or knockout condition. There are several base pair edits in this SDN2, and it is gain of function mutations can occur. In third, there is insertion of foreign gene or DNA, and that's why it has not been approved yet. It is considered like that of we will say transgenic or genetically modified crops. So here there is gain of function with new protein is possible. And finally, I will talk about crops with novel traits. Just a minute. Okay, so what is meant by crops with novel traits? The traits which were not earlier available in crop system, those can be incorporated. So this is one. If we eat rice, okay, there is no vitamin vitamin A which we can get from rice. But this person, Ingo Putricus, okay, he decided to incorporate this particular provitamin A into rice, and from its color it has been called as golden rice. Okay, these are two scientists from India who work with this Ingo Putricus, and they have developed this golden rice. So what is there? In this case, this white rice, okay, with these three genes, phytoin synthase, then PDS and cyclase, lycopene cyclase. With these three genes, there was incorporation of beta carotene in the rice seeds. Okay, so further these transgenics have been again incorporated into our Indian varieties, and this Swarna, golden Swarna has been developed. So you can see this. This is simple rice with the incorporation of three genes. Now there is provitamin A, and with this provitamin A, it can help to develop, we will say, vitamin A rich rice. And what is the use of this? In country like India, okay, if we provide this, it can solve the problem of night blindness, which is there. And again, it is in the process of releasing in India, but there are some problems. with copyrights and all those things there, but it has been released in Philippines. And now we are witnessing, we will say, utilization of this particular rice. Then if we incorporate few genes, we can increase beta carotene up to 400 folds in maize, or ascorbate 6-fold, or we will say 2-fold increase in folate. Then there is again novel varieties like edible vaccines. Okay, where pathogen protein can be cloned and it can be incorporated into either potato, banana, tomato, or any other edible fruit, or we will say vegetable. And like this, we can get immunized rather than getting injection for diarrhea, hepatitis B, measles, rabies, or nowadays we will say for even for corona. Then this is very interesting, even it is not related to, we will say, agriculture, but very important and very interesting example. We have seen that during Second World War, about 100 billion, or sorry, million unexploded landmines are present there, and because of which every year around 26,000 people get killed while working in that area. So in this case, the gene has been incorporated so that when you plant this material in that area, because of nitrogen oxide which is coming from evaporations of these explosives, the plant get convert into purple, from green to purple, and we can identify, yes, there is mine, and we can further take out that unexploded landmines. So like this, it is helping farmers to exploit new areas. Again, we know that there is no blue color rose, but with the help of, we will say, or violet color carnation. So with the help of these molecules like 580 or BP40, we can develop this delphinidine, and we can get this blue color or purple color flowers in the industry. Okay, so there are more than 32, we will say, crops which has been grown. And I will say this is very important, how you are relating these transgenics with sustainability. So here we can say there is increase in crop productivity, there is conserved biodiversity with this one, and provide better environment by decreasing herbicide and insecticide use. And again, most important is there is reduction in carbon dioxide emission. And how we will calculate this one? Like taking out 16.7 million cars from the road for 1 year. So that much pollution which is created by almost 17 million cars every year. So this much carbon we are reducing because of using these transgenic crops. And obviously it is helping to elevate property and hunger. Okay, so I will say we have started with normal selection, and now we have reached up to golden rice. So with molecular approaches, we are using this one for sustainable agriculture. And it is not, we are sequencing just few crops. Now there is project called Earth Biogenome. So what is it means? By our genome sequencing technology, informatics, automation, or artificial intelligence, we are able to understand, utilize, and conserve this biodiversity in proper way, in sustainable manner. And it can, again, efficiently use this particular genome for our better agriculture. So I will say in my last slide that molecular approaches through agriculture biotechnology can increase higher yield, it can provide food security, it can provide better environment. But for me, or for everyone, I will say sustainability is real issue, because through science and through ethics, we have come to the realization that we are bound by the law of nature and we must obey those laws to make agriculture and our civilization more sustainable. So I will stop here. Thank you very much.
Thank you, Dr. Solanke. So now we can take up questions. And few people, participants, they have posted questions. So we can start from that one, from the beginning, so that we'll not lose the track. And if someone is willing to raise hand, they can do that. So why conventional breeding is shifting towards modern technique? As some scientists say that consuming GMO can change human genome also. Is it myth or not? Two questions together.
Okay, there are two questions. Why we are shifting towards modern agriculture? We have seen Bengal. That's why I have in my introduction I have added this Bengal famine. So which means when I think in 2016 I was fortunate to meet Norman Borlaug sir, and some reporter asked him. He came here in Delhi for some fertilizer company. And then someone asks you why you are not growing crops on in organic way. So he told, I don't have problem to grow crops in organic way, but from where we are going to get this much organic material to put it into soil. And we know that our soil is there is decrease in all type of micronutrients or macronutrients. So one or other way we need to add. The problem is that we are adding, we are only concentrating only on NPK, and we are not able to see this other part of the macronutrient. So if we are able to provide this kind of organic material, definitely it will be good. And sustainability is very important. Until and unless during, we will say, green revolution, we lost that sustainability part of the agriculture, and that's why we are facing this. But with new technologies, if we are able to make agriculture sustainable, I think that will be the good option. Sir, what was second question?
Second
Part of this.
scientist myth means consuming GMO can change human genome.
No, we are consuming, or we will say, forget about us. Americans are consuming, we will say, soybean oil, then all soybean parts like protein. They are eating maize, they are eating brassica, but there is no single issue till date which has been published with authentic information for the change in genome because of this one.
Just for the information of participant that we have posted link for the feedback in the chat box. Everyone is requested to fill that feedback form while we are discussing. It takes just few minutes. Second was golden rice available in their market, whether this golden rice or BT brinjal of why it is not commercialized. We can address together, I think.
Yeah, so basically, golden rice maybe within few years it will enter in Indian market. Since this is transgenic, okay, it is not only scientist, we will say, clarification. In India, country like India, people perception is also important, and probably we were not able to present our science in proper way in front of, we will say, all people. Okay, we have restricted ourselves to our labs, or we will say few conferences where we are telling. So if we go to all people, and we were able to tell that what is there and how this particular has been developed, there will be no issue. And if we talk about BT cotton, there was moratorium in 2014, which has been it was for 10 years. After that, GAC, it is there, but it was released in Bangladesh, and we will say they are consuming it, and there is no problem. So it means now we have data from at least our neighbor country that there is no single issue because of we will say BT brinjal. So I think you people can support us for releasing this in India. So it is since it is public perception, we need to change this perception for transgenic crops in India.
Can we learn more about genetic fidelity testing and how to get certification? Your favorite.
Sir, I think you are better person to answer this question.
I would request you address that.
Okay.
A little bit you can cover primer also, markers, that ISSR primer, why it is so relevant. So I think this would be good learning for them. Just take 2-3 minutes to explain that ISSR marker and suitability. We have been working on all the aspects, but you explain that.
Yeah, sure. So see, if we talk about genetic fidelity, it means whatever material we are getting from its mother plant, it should be exactly similar to its mother plant, what is going to the field. And why it is important? So for example, if we have taken Grand 9 variety and we are expecting that it will provide us 40 kg of banana from single plant. Okay. And if there is no genetic fidelity because of tissue culture process, if there is variation in its genome and this next plant may be sterile or it's very dark, it is not, or it is giving, I will say in terms of commercial point of view, just 20 kg or 10 kg of fruits. So it means it is not good. Farmer has purchased that particular plant considering that it will provide 40 kg. So in this case, by genetic fidelity with few markers, and again we are using here ISSR and IRAP markers because these markers can provide the variations which supposed to be not there in genome. And again, because of tissue culture, there is change in ploidy level or there is transposable elements get increased, and those can be identified with these markers, and this will help us or this will help to protect our farmers to get we will say which the plants which are not true to type. So like this markers can help for genetic fidelity. And how we can do it or how we can get certifications. So basically, if there are companies, those can register with this, we will say NCSTCP. If you are, if someone from you are from company, and you can visit the site of NCSTCP, there you can get more information for this one.
Just to supplement, companies should be in commercial mode, at least 0.5 million capacity, 5 lakhs plants. The capacity, not production. You can produce less also. And after recognition by Department of Biotechnology, you need to get your initial planting material tested for the buyers. And whatever progeny, the batches you are producing from the plant, the mother plant, that batch has to be tested by random sampling, normally 0.1%, depending on the size, it varies. And that has to be tested by testing laboratory authorized by Department of Biotechnology. Then you get certificate of quality and a label. And the label has that QR code. So consumer will get label with QR code, and this is traceable. Which company produced, which variety, when it was tested. So complete traceability was there. I still remember 2012 to 2016, we were working on the all traceability aspect. And by 2015-16, we could complete that web portal with all the traceability. So that is the broad coverage of certification for the information of any startup or company. Now, can CRISPR-Cas based plant be commercialized? No, no. Before that, we
Uh,
have a question from Dr. Chanchal, it seems. From QTL, how we can pinpoint single gene for editing?
okay, you are mixing two things, Chanchal. Actually, QTL is there is no need of editing. We can edit only those traits or those genes which have negative effect. QTLs are mostly we select QTLs for positive effect. And from QTL, if we do fine mapping, we can identify, because if we say QTL, okay, so QTL means the range from one marker to another marker, in which there may be uh, 10 genes, 20 genes, and we can slowly narrow down that particular range and further identify one gene by which is one, or we will say few genes which are related to that particular trait. And after that, we can prepare gene-based marker directly, and using that gene-based marker directly, we can use it for molecular breeding. Okay, genome editing only we can do if that particular QTL have negative effect.
Now, Dr. Priyanka is asking, can CRISPR-Cas based plant be commercialized?
Yes, I told that there are three type of SDN1, SDN2, and SDN3. 3 years back, Government of India gave notification that SDN1, where there are few mutations, or we will say, or we will say some SDN2, where are more number of mutations in particular gene. So here gene should be already present there in that genome, and if we are mutating that, okay, so these two can be commercialized without all these restrictions of we will say GM crops. So there are means we have to go through some process, but there is no need to for these restrictions like which are there for these GMOs. So we can maybe within, I think 1 or 2 years, we can get 100 of we will say uh, varieties with genome edition in India.
Thank you. We have a few minutes more to conclude. I hope all the participants, they must have filled that feedback form. If anyone is facing difficulty, they can comment in the chat box. So, Dr. Nilu Joshi is asking, what is the status of edible vaccine, its commercialization part?
Okay, till date, edible vaccines are only in laboratories.
You have to see
Uh,
the pain point.
Yeah, so it is yet to be commercialized, but since slowly everything is coming to market now, maybe if somewhere if we are able to see this, uh, but in coming few years, I am not able to see any product of edible vaccine, or we will say in market. So, but right now it is their work is going on in laboratories for that one.
We have placed this feedback link in the chat box. So please use that chat box link for the feedback form. We are not placing in the group. Now, Mr. Pratik Balaji asking, what is the better route for protecting IP in this domain? PPV4A Act or patent? Means he's asking for the variety protection or patenting the gene. Which would be better for the IP part?
In India, we cannot patent gene or even plant. So we have to go by this PVV FRA route only, because in this case, at least the breeder who develop that particular variety, he can get benefit from this one, because patent is not helping. Single gene cannot be. It's we will say gene and plant cannot be patented in India.
Unless until it is modified. If you have modified something created new, then the modified one can be considered. But here recommendation is that variety registration would be preferable for the
As
seed point of view.
Mhm.
Can we use EST SSR marker for genetic fidelity?
No. If we talk about EST or SSR markers, so those are the fixed variations present in varieties. So we cannot expect variation in specific, we will say SSR marker because of genetic fidelity. As because of, we will say tissue culture, there is variation in its ploidy level or repetitive regions or in transposable elements. So SSR markers are not recommended for genetic fidelity testing.
Any more question? All participants, they have submitted feedback form. Admin can just unmute everyone, give the right to raise their hand and to talk before we close. Siddharth?
सर दे दिया सबको राइट.
Okay. So it's open forum now for 2 minutes.
Sir, what is the constitution of this group, sir?
This group basically it is a faculty development program, so we have lots of participants from the academia, ranging from scholar to professor.
Okay.
Then we have industry participation. There are many industry that are participating like Ajit Seeds and many more, Jagdamba Bioplant, other company, Seal Biotech. And every day we are trying to keep one scientific lecture, one commercial part. Like today we had a biofertilizer, now you are talking. Tomorrow we'll again talk about gene editing part.
Okay.
And then the technology valuation and commercialization part. Last day we'll have secondary agriculture and talk on the Kumar bioplant, that is floriculture.
Okay.
So every day we are balancing like 50% industry partnership.
Mhm.
And the two experts from abroad.
There is one query. Are any kits available for virus indexing? Yes, there are virus indexing related kits are available in market, but mostly those are used by tissue culture companies or means who are doing like accredited testing laboratory right now. But for many crops, this virus indexing kits are available in the market.
Kits are available or many
Sir?
times PCR
Sir?
based detection is being done.
Hello?
Yeah, Manish
Sir,
sir.
there is a one question. Sir, what is the procedure for releasing a variety? If you want to release, what are the steps? What is the certification program of it?
Okay, so in this case, if we have variety, we have to submit it to where you want to release. Okay, if it is for state or if it is for nation. Okay, on the basis of that, we need to submit it. Then it goes for that particular area for growing continuously for 2 to 3 years. And after that, it get compared with check. Check means the already established variety, and if it is yield is more as compared to that, then only it get released.
Normally, it's a dust test they
Sir,
perform.
And
Sir.
it also
US.
goes through All India
Hello.
Coordinated Project trials also.
Okay, sir. Okay, thank you, sir. Thank you, sir.
Dust test. They call it DUS, dust test. Distinctness, uniqueness, and stability. It has to be stable when they are growing during trial phase. Okay. So if anyone face problem in filling the feedback form, they can also write in the group. Then we'll paste in the group also so that you'll get time to complete your feedback form till 10:30. Thank you so much. So this is time to close. Exceeding the time little bit. Dinner time for everyone. Many might have completed while closing the camera. It's good to complete that on time. If not completed, then have your dinner. And once again, thank you, Dr. Solanke, for this wonderful presentation
Thank you very much.
on behalf of KSU, Kausalya, the Skill University, Government of Gujarat, and on behalf of all the participants from academia and the industry.